Hello to all students. In this video lecture, we are going to discuss types of RNA. To understand the concept of types of RNA, we are also going to discuss a topic which is known as how cells synthesize protein. To understand the process of synthesis of protein in our body, first of all, we have to know that there are so many different types of proteins in our body. Approximately 10,000 different types of proteins are present in our body, which include muscles, enzymes, hormones, hemoglobin in the blood, antibodies, antigens, similarly hair, nail, which is a keratin protein, and so many other types. These proteins are synthesized in our cells. There are trillions of cells in our body. So I am going to take one cell an example how the cell synthesize different types of protein. So let's go to the factory of protein. So we are going to take an example of a cell. This is a diagram of a cell. So let's consider this cell as a factory, and this factory is producing, synthesizing different types of proteins every day, 24 hours, 7 days a week. A factory should have an office for proper working of the factory. So this is the office of the factory, which is known as nucleus, and each office has a revolving chair, and the revolving chair is known as chromosome. So there are 46 different chromosomes present in the nucleus, but I have shown only one nucleus, one chair. And factory is incomplete without a boss, and the boss is DNA. DNA contain all the information relating to all the proteins present in our body. It has special information, special codes, special genetic information which is stored in the DNA very secretly, and it has been protected so many ways. Suppose the boss, which is DNA, receives a call from the follicle cell which is producing hair. Follicle cells calls DNA that I need 10,000 molecules of keratin protein so that I can increase the length of the hair. As soon as order is received by DNA, DNA start the process of synthesizing protein. It is obvious that the DNA is not going to go into the factory and synthesize the protein by itself. Rather, it has three specialized managers. These managers are known as RNAs. The most important managers are messenger RNA, abbreviated as mRNA, transfer RNA, abbreviated as tRNA, and ribosomal RNA, abbreviated as rRNA. The most important one is messenger RNA. Messenger RNA is 5 to 10% in the cell. It is a single-stranded molecule of varying length depending upon the type of protein. For example, if a protein has 1000 amino acids, the length of the transfer RNA will be 3000 nucleotides. These nucleotides arrange along the length of the messenger RNA in a special sequence in the form of codons like GAC, UCG, AAU, etc. This messenger RNA has two ends, 5' and 3'. 5' is protected by 7-methyl GTP and 3' end is protected by poly A tail. Messenger RNA contain all the information which is coming from DNA. So this information is messenger RNA. The most important function of messenger RNA is to transfer the genetic information from DNA, which is present in nucleus, to the ribosomes present in the nucleus for the synthesis of protein. So as soon as the order is received, the DNA is going to call messenger RNA, and then through a process, very important process known as transcription, DNA is going to transfer genetic information, specialized information for the synthesis of a particular protein, keratin for the synthesis of hair, and those genes are transferred from DNA into messenger RNA in a process known as transcription. Now that all the information has been transferred into messenger RNA. Now this information is going to be protected by 7-methyl GTP cap on 5' end and poly A tail on 3' end, so that messenger RNA while traveling does not lose the information present on the messenger RNA, or enzymes cannot dissolve the messenger RNA, because this information is very sensitive. Now this messenger RNA is going to take this information from nucleus through nuclear pore into cytoplasm towards ribosomal RNA, where information is going to be decoded in the form of protein. Second type of RNA is transfer RNA. As its names indicate that it transfers amino acids. There are 20 different types of amino acids present in our body for the synthesis of different types of proteins. So there are 20 different types of transfer RNA, each for separate type of amino acid. I have shown right over here 3 transfer RNAs. It has a base which has anticodon, it has different arms, and I am going to discuss its structure in detail later in the lecture. And it has amino acid on the top. Each transfer RNA has different amino acid attached with it. And these transfer RNAs are going to transfer these amino acids to ribosome for the synthesis of protein. And the third type of manager, which is very important, is known as rRNA, which is 50 to 80% in the cells. It is composed of 2 subunits, larger subunit and smaller subunits. These subunits are present in the cytoplasm freely, dispersed in the cytoplasm, or they can also be present on the rough endoplasmic reticulum. These ribosomal units are going to synthesize protein in a process known as translation. Messenger RNA has been arrived where the ribosome is. Messenger RNA is going to attach with the ribosome. A ribosome is going to read the information present on the surface of messenger RNA, which is present in the form of genetic codes like G, A, C, U, C, G. These codes are translated into proteins. But first of all, a ribosome is going to read AUG. This is a code, and the anticodon for this is UAC. Anticodon is present on the surface of transfer RNA. This transfer RNA will move from cytoplasm towards ribosome, where it attaches with the larger subunit of ribosome. Then second code is read, and then second transfer RNA is called. When 2 transfer RNAs attach with the larger subunit of ribosome, their amino acids attach with each other through peptide bond right over here. And then these transfer RNAs are released from the ribosomal RNA. And the next codes are read then, and new transfer RNAs are brought in. And each transfer RNA giving amino acid to the growing polypeptide chain, growing chain of protein, growing chain of amino acid through peptide bond. After all the information present on messenger RNA has been read and decoded, a protein molecule has been synthesized. This protein molecule can be of varying length depending upon the type of protein. Some proteins are short, some proteins are large and very long. After the synthesis of protein, this protein is transferred to the follicle cells, where hair is going to be increased or hair is going to be grow, and next day you will see that the hair has come out from the skin approximately 1 millimeter, because the protein has been attached in the follicle cells in the follicle and the hair has been grown. Similarly, nail can grow, our muscles can grow, our enzymes, our hormones can grow, and every day thousands of different proteins are synthesized by transcription and translation, DNA and its managers, 3 different types of RNA working continuously to produce different types of proteins. So to produce good protein, we need amino acids. There are 2 types of amino acids, essential and non-essential. Some of them our body can produce, which are non-essential, while some we need from the food. The best source of amino acid is meat, beef, mutton, chicken, fish. They contain different types of very essential amino acids which we will take through our diet, and these amino acids will help to build different types of protein in our cells. I hope it makes sense. That's all for today. See you in the next lecture. Until then, bye.